Okay, this little mini lecture is going to introduce you to, some of the techniques and a little bit of background for the Varicella Zoster CREB paper. So VZV stands for Varicella Zoster virus. This is in the alpha herpes virus family, and though there is not a specific wiki on this, Epstein-Barr virus and HSV1, herpes simplex virus 1, are in this similar family, I believe. Anyways, there are herpes viruses. I don't know if they're alpha or not. So you can look at those wikis for a little bit of additional information. You can see that this is an enveloped virus, polyhedral capsid. So here's the capsid. VZV has a double-stranded DNA genome, approximately 123,000 base pairs, so kilo base pairs. VZV's genome encodes about 71 proteins. And we'll talk about one of the mutants. So all of these numbers stand for open reading frames, ORF, open reading frames. So those are equals protein coding. So you'll see that they're going to make a VZV ORF10 mute or a GFP fusion. So that's what the 10 means. I'm just kind of looking here. There's a ORF66. So this is one of the kinases. And there's an ORF47 right over here. So this is another kinase that we'll talk about. So this is just to help you kind of understand what's going on. And so you can see that they've characterized some of these proteins, whether they're dispensable, meaning that the virus can still replicate without them. They help in tissue tropism, so the specific cell types that the virus can infect. Mutating the ones in orange produce a severe growth defect, and the red ones are the essential. So lots of proteins encoded by this virus. And again, Epstein-Barr virus, EBV, and herpes simplex virus 1, HSV, if you look at those wikis, it will give you a bits of information about how this double-stranded DNA virus replicates. And I meant to say it's also a linear genome, so it's like a big chromosome. Okay. Other interesting and really important point about VZV is it makes a latent infection. So Varicella Zoster, a long time ago they thought that chickenpox was caused by one virus and shingles was caused by a totally different virus. So varicella was the chickenpox virus, zoster was the shingles. Now we know it's the same virus. It just shows up at different times in your life. So chickenpox is really the primary infection, and it's usually in younger people. You may not know, but chickenpox is a vaccine preventable disease. Zoster, so what happens in this latent infection is the VZV virus hangs out in sensory neurons and then can become reactivated. That's the definition of latency later in life. So usually older people, usually greater than 50, I think, sorry, maybe it's 60, but it can happen in anyone. The biggest issue with shingles is this post-herpetic neuralgia. So you get this, you can, get this intense pain, this disruption of these neurons, very long lasting, like years. And so this is the main reason that we also have a vaccine for zoster. So the shingles vaccine when you're, I think it's over 60. I don't know. I'm getting up there, but I know they haven't told me to get it yet. And it's not just that the shingles is so bad, but it's this potential post-herpetic neuralgia that's really a long-lasting complication. Chickenpox and shingles, the two different diseases, are shown here. Again, children get a chickenpox vaccine to prevent chickenpox, but the vaccine is live attenuated. Okay, so that means it's a live vaccine, which means it can stay latent and reemerge as shingles, even though it's supposed to be attenuated and not cause disease. So I really thought this was interesting that we weren't able to make an inactivated chickenpox vaccine to protect you against both chickenpox and shingles. So I'm just checking my notes here. I think that's what we got. And so you can see chickenpox will go throughout the body, and shingles usually comes on the side, sometimes on the neck area, back, shoulder area. So it's really being activated in a neuron that's targeting these epithelial cells. So this paper is about the role of a host protein, CREB. Okay, so CREB stands for cyclic AMP, response element binding protein, which CREB is shown down here. CREB is a transcription factor, which means it's important in turning on gene expression. And this gene expression is coming through lots of different cell signaling pathways. So prior to this paper, they did some screening and identified CREB as a host factor that was altered in expression levels during VZV infection. And what they found is CREB phosphorylation. That means it's getting a phosphate group added, is increased during a VZV infection. And they also know from their previous work that phosphorylated, and I'm going to write it as p-CREB, phosphorylation, is important or required, I should say, for a productive VZV infection and cell-to-cell spread. So in order to get virus made, replicated out of the cell, spread to the next, you've got to have phosphorylated CREB. And remember that the enzymes that do phosphorylation are called kinases. So they're the enzymes that put phosphate groups onto a protein. The other molecule that will, P300 CBP, P300 CREB binding protein is another protein that CREB interacts with to help initiate transcription. So that's a little bit of background about the proteins we're looking at, and the virus we're looking at. This is a little bit of information about the techniques used. So in figure 1, 3, and 4, they'll talk about flow cytometry. And what flow cytometry is it's a way to count cells with specific characteristics from a mixture of cells. So what this diagram is trying to show you is you can have a heterogeneous population of cells, and what we do is we use antibodies that are specific to something expressed in that cell. So those antibodies tag the cells, and those antibodies will have different colored, fluorophores on there, right? So we've talked about antibodies last paper, so you recognize the structure, and then we can tag them with different colors like red and green and blue and yellow and all these variations. So then you put this mixture through, there's a laser, and it can actually count, okay, there's one red one, there's one yellow one, there's one blue one, there's one green one, you know, whatever antibodies you have. You can actually do something called FACS, which you can actually sort these cells into little test tubes that hold the different groups of cells. But in this paper, they're mainly counting. So you're going to see data, like these graphs. And what this is showing you is the count, which is the number of cells. And across the bottom is where you get different fluorophores or colors. Sometimes they call it different intensities. So, blue, say, is going to have a certain intensity, and red will have a different one, and green will have a different one, and yellow will have a different one. And so you get these graphs where it's counting the cells, and it's counting them at these different intensities as it's flowing through, and it plots it all here. So then you can look at basically different populations. And so yes, with the antibodies we have, you can differentiate between CREB and phosphorylated CREB. Okay, so you're going to see that they're going to look at phosphorylated CREB, infected cells. Let's go to the next one. They're using fibroblast cells. This is a picture of them in tissue culture. I think these are HELF cells. For all these papers, don't get bogged down in the types of cells. Okay, so what's important in this paper are the proteins that are being expressed at what levels, and how much virus is being produced. The cell type is just the cell type they have that works in the lab for VZV to infect. Okay, so don't take any significance out of fibroblast cells, because if you looked up VZV, it's really infecting epithelial cells and some neurons. Okay, so this is just for tissue culture, in vitro, and the challenges with studying viruses in vitro is a lot of times you're not able to use the actual cell types that the virus naturally infects. And so sometimes what we have found in the lab doesn't always translate to humans. Okay, in figure 1 and in figure 8, they use something called immunofluorescence. Anytime you see the word immuno, it means antibodies. Fluorescence means you're going to get some kind of bright light that you can detect. And so what's happening is you have a primary antibody that is protein specific. So when we talked about the influenza paper, we talked about antibodies specific to hemagglutinin of influenza. So we can have primary antibodies that are specific to Varicella zoster virus. We can have primary antibodies specific to CREB protein. We can have primary antibodies specific to phosphorylated CREB. All these different proteins. Okay, and so what you're doing is the antibody will bind. So you have something very, very specific happening. Then you have a secondary antibody that's linked to something that fluoresces. And this is where we have different colors. So this diagram down here is just trying to show, for instance, you could have a red fluorescent and a green fluorescent, right? And so you could be detecting different specific proteins being expressed in these same cells. Okay? And this is what's called a merge. And you will learn in immunofluorescence microscopy that red plus green makes yellow. So wherever you see yellow, that means both proteins were being expressed. Both proteins were being detected. The other kind of staining is Hoist staining, which stains the nuclei. So here it's seen nicely. You can see the whole cell and the blue nuclei. But normally what we see in fluorescence is just the blue. And you can see that also in these images. So you can see the blue staining the nuclei of the cells. Let me make sure. Okay. Then we have one other kind of fluorescent molecule, and that's GFP. And that stands for green fluorescent protein. This was a really cool protein isolated from some jellyfish, and it fluoresces under UV light without having to use antibodies. So no antibodies needed. So the cool thing is that they've made a Varicella zoster virus that's recombinant, so they've genetically manipulated it to have green fluorescent protein expressed whenever the protein from open reading frame 10 is expressed. Basically what that does is wherever you see green, it means VZV is replicating, it's expressing its proteins, right? And here this nice black screen is just uninfected, so you don't get any background. So here you can actually skip all the steps of fixing the cells and using antibodies and fluorescent markers, because wherever the virus is replicating, those cells will glow green. So let me stop and just give you a little orientation to some of the viruses, strains you will see. So POKA, this is the regular old VZV virus, and this is, virus, and this is the vaccine strain. Okay. So everything is derived from POKA. And honestly, I don't know what the OKA means. I tried to look it up, but it just says it's the original virus. The parental is what the P stands for. Okay, so we have one, we talked about in figure 1, that has GFP fused to the open reading frame. We have 3 kinase mutants. And remember that I said to get phosphorylated CREB, you need a kinase that adds the phosphate. So one of the questions they were asking was, does one of the VZV kinases, or no, let me rephrase that. Is one of the VZV kinases required for phosphorylation of CREB? Because if they see an increase in CREB phosphorylation in VZV infected cells, you might think that the virus is responsible for this. So they've made point mutations that have knocked out the kinase activity. And they're asking, does this affect CREB phosphorylation levels? And I think, as far as I can tell, the chimera is a combination of these two. So you can knock out one, you can knock out the other, you can knock out both. I'm honestly not 100% sure, but that's what I could glean from this paper. The other VZV strain they'll talk about is this Oka R6362FL. This one has Luciferase. Okay, so Luciferase is also not a fluorescent protein, but a protein that produces a signal that can be detected. So Luciferase is actually originally isolated from fireflies. So the little glowing firefly butts, they've isolated that protein. And so now they've made a recombinant virus that expresses Luciferase, and these are going to be used, like I said, in figure 5 in the in vivo mouse experiments. Luciferase is nice. It makes a really bright signal when you add in a substrate called luciferin. Luciferin. It makes luciferase glow. Okay, and you can detect it, and it's bright enough to detect in vivo. So the cool thing is you don't have to sacrifice the mice to look at, say, green fluorescent protein or antibody staining. Under the microscope, you're going to see that they can take live imaging. But yes, they have to inject this substrate into the mice to react with the luciferase and make it kind of glow. And it's not like you see glowing mice, but you have imaging equipment that can detect it. Okay, Western blot. Very common molecular protein technique to detect specific proteins. Now, this is not used in the cell like immunofluorescence. This is taking samples from the cell. You separate all the proteins from a cell, and this is called an SDS page. Okay. And it really should show you like hundreds, thousands of proteins in every single sample. And then you transfer from the gel, you transfer the proteins to a membrane. So the membrane is kind of like a piece of paper, different material, but gels, if you've ever worked with them, gels are very flimsy. So you cannot manipulate them very well without breaking them. So we can transfer the proteins to this membrane, and then we take the membrane and we do something kind of like immunofluorescence. So you have proteins attached, you have a specific antibody, and then you have a secondary antibody that gives you a signal. And so what you would see on your Western blot is the signal that says, okay, in these samples, this is the specific protein. And it's a great way to quantify or get a measurement. So when you're doing this immunofluorescence, it's more like it's here, it's not here. You're not getting a great quantification. But with Western blots, you get specific proteins and you get to quantify them. So two of the main antibody types they're using, phospho-CREB will detect only CREB protein specifically, where that's interesting. Why is it called phospho-CREB? Oh, no, sorry, this isn't phospho-CREB. Oh, just CREB, sorry. So we have an antibody that's just specific for CREB, and an antibody, sorry, that wasn't too good, specific for the phosphorylated version of CREB. So you can quantify how much non-phosphorylated, how much phosphorylated is there. What you're going to see in the Western blot samples with the phospho-CREB protein is it picks up some additional bands. So one of the main other proteins this antibody picks up, detects, is a phospho-ATF1. So I just want you to understand when you see other bands of different sizes below the phospho-CREB, those are non-specific binding. It's very challenging to get antibodies that bind to very specific proteins, especially if you're trying to differentiate between a phosphate group or not. So the antibody's also picking up phosphate groups on other proteins. So that's why you'll see multiple bands. The other thing I want you to understand is what's called a loading control. So if we're going to quantify or quantitate, and we're going to say this sample has more or less than this other sample, well, you have to make sure that you've put in the same amount of protein for each sample so it's a fair comparison. And that's what these loading controls are. So the two loading controls that are used in this paper are alpha-tubulin, and this is just for cytoplasmic proteins, and lamin AC for nuclear proteins. Okay, and you want to see that it's about the same amount of your loading control. So if someone just showed me this data and said, oh yeah, look at this sample, there's a lot more, I'd say prove that you didn't just load less into sample 1. That's the point of the loading control. And what you actually do is you take pixel images of this compared to this and you do ratios to show the actual difference. So we need two different types of loading controls because we're looking at two different parts of the cell. XX65023 is an inhibitor of CREB. So if we think CREB is important, what we want to do is inhibit its action to test if it is truly important. So they've found the small molecule that disrupts CREB-driven gene expression. It blocks the CREB CBP interaction. So it's actually working on phosphorylated CREB, which it doesn't specifically say. Okay, so here's a tool we have to say, okay, well, if CREB's important, what if we block it? What happens to the levels of virus VZV infection and production? Plaque size is another interesting technique they're doing to look at how much replication is happening. So I've talked in other videos about plaque size. And remember, what's happening here is you have a virus that infects and it makes more virus and it spreads to the new cells and it affects those, and then it makes more virus and it spreads and it kills, cytopathic effect, and you have this clearing. So normally what happens is all the purple here are the live cells, and the clearing is due to a virus. Okay. Some viruses naturally have different plaque sizes. So that's what this picture is showing. This is a mixed virus population, and they're looking at plaque size as an indicator of what virus is infecting. In our paper, they're looking at plaque size as an indication of how good replication is. So you could imagine that if I took this diagram and I erased this outer circle, if there was not a lot of virus replication, I'd have a smaller plaque than if there was a lot, right? They're doing a different technique than I've seen before. So VZV must not make nice cleared plaques. And what they're doing is they're using an antibody stain. So all these little pink things, here they are in close up. And they're staining for VZV virus. And they're looking at how big the VZV spread is. So it's the same concept. They're calling it a plaque assay, but it's not a clearing. They're actually having to stain for the virus there. Finally, and kind of like we discussed in the last paper, do things that work in mice translate to humans. VZV is a very human-tropic virus, so it doesn't infect lots of different organisms. They said it does infect some guinea pigs, but what they've developed is a mouse model. So SCID mice, these cute little mice, severe combined immunodeficient mice. So they have no adaptive immunity. So they don't make antibodies, they don't make the cytotoxic T cells. So it's kind of a nice model to look at, in this case, a virus infection, because you don't have to worry about the immune system knocking it out. You can see what is the virus doing in something more in vivo-like than fibroblast tissue culture. So they've actually grafted human skin, fetal tissue, onto these mice, and then they can infect them with virus. So this is in vivo. And remember, if you write in vivo or in vitro, you have to put it in italics. Since I can't write in italics, the other option is to underline it. And so they're going to look at kind of a more realistic model for what is happening with a VZV infection and what is happening when you add this drug. So something we've talked about in class is antivirals. So maybe they're looking at XX molecule as a potential antiviral. So here's where that last virus strain, let me see where I wrote it, is. So they're using recombinant OKA R6362FL. So this is the Luciferase expressing VZV virus. And so what they can do is basically, in this case, this is just a really general picture, they're using a virus that expresses luciferase. And then they're imaging the intensity of the luciferase protein expression, right? So the brighter the red, in this case, more virus being expressed, more luciferase being expressed. And again, the cool thing is you can look at the same mouse over time. You don't have to sacrifice the mouse. And if this was a VZV infection, you can see how it's spreading. Okay, I think this is the last one. Final technique they're using in figure 7 is H&E staining. This is just a very common histopathology staining, so they kind of want to look at what do those skin grafts look like. Hematoxylin stains the nucleus, eosin stains the cytoplasm. So they're not necessarily looking at virus infection, virus production specifically. They're saying, what is this virus infection doing to the cells, right? And they're comparing in here untreated DMSO, which is your vehicle, so that's just kind of your other control with just the injection but no virus, and this is with the drug XX65023. Okay, so you'll take a look at these and kind of understand a little bit about what's happening to the virus infection in the presence of this drug. All right, I believe that is all I have for you. I hope this helps you work through the VZV CREB paper. Different than the last paper we looked at. Now we're kind of looking at host cell virus interactions. Okay, thanks.